Thank you, Daniel, for an excellent talk of a very complex subject area, and I think you explained it very well. Remember, folks, that you can put any questions that you have for any of our speakers in the Q&A section. We're going to round off this day with Dr. Anna Bibby, who is a consultant respiratory physician from North Bristol NHS Trust and also a clinical academic at the University of Bristol, and has been doing excellent research in both pleural disease and mesothelioma over the years. And she's going to be talking to us about assess meso. Thank you, Anna.
Thanks, Selena, and thank you to Kevin and the organisers for inviting me to give this talk. Great. So yeah, I'll just run over a general overview of assess meso, which has been mentioned a couple of times throughout the afternoon. I'll give you an idea of why we set up the study in the first place and what we're hoping to achieve with it. A brief update on where we are with it, and then spend the latter half of the talk talking about the trials and sub-studies that we're embedding within assess meso. So currently, everything we know about mesothelioma in the UK comes from these two reports, which is from the Health and Safety Executive of Great Britain and the Mesothelioma UK National Audit. And then also, obviously, we've heard about the really very important, groundbreaking trials that have been published in the last few years about new treatments for mesothelioma. Now, these reports, these documents, these studies are all incredibly valuable, not least the trials because they've offered us new treatments, but they do all have certain limitations. So the HSE data is mortality data, so it is essentially reported post-mortem, which as a clinician, in fact, I think if anyone involved in mesothelioma would say is too late. It is a snapshot in time, as is the Mesothelioma UK report, although it does track people through their clinical diagnostic and treatment journey. But they're not patient-focused. They don't give you a sense of what an individual person has been through in their journey with mesothelioma. When we talk about clinical trials, and we heard earlier about the challenges of running robust clinical trials, and they are challenging. And it is almost by necessity that a phase 3 randomized trial of a drug has to have eligibility criteria. And that is important to ensure the trial provides accurate output. But that means that the people who have gone into the trial are not necessarily representative of the wider population. A couple of particular examples of that are that for most clinical trials, you have to have either had chemotherapy already or be willing to have it as part of the trial. And we reported from our unit a couple of years ago that about a third of people who are offered chemotherapy choose not to have it for various reasons. And so those people are automatically not eligible for the trials, but also it means that they are not represented in the literature. They're not reported. Their data, their story is missing. Similarly, peritoneal mesothelioma has historically been somewhat neglected in favour of pleural mesothelioma based entirely on numbers. But again, as a result, people with peritoneal mesothelioma are less well represented in the literature. There's a caveat to that, which is the recent confirm study of nivolumab, second-line nivolumab by Dean Fennell, included people with peritoneal disease and should be commended for that. So just to demonstrate why it matters that people in trials are not necessarily representative, this is a paper that was published last year from a group in Australia, and they went back and looked at all of their patients who had come through their clinic doors over a 5-year period. And they compared their patients with the eligibility criteria of two of the major clinical trials, the bevacizumab trial, MAPS, and one of the pembrolizumab trials. And they looked to see how many of their patients, their clinic patients, would have been eligible for the trials had they been running at the time. And they found that only 50% met the criteria to go into MAPS to have bevacizumab. And although slightly more met the eligibility criteria for the pembrolizumab trial, it was still less than 2/3 of their patients. So, yeah, half to 2/3 of people are going into or could go into these trials, but that leaves a huge population that can't. Now, the next slide is a survival curve, so do look away if you don't like that, but it's brief. Why does this matter? And again, this is just demonstrating the difference in characteristics between the two populations. The red survival curve is the people who were not eligible to participate in trials, and you can see that their overall survival is much shorter than the people who did participate in the trials. And you might say, well, that is obvious because the people in the trials got these drugs. And yes, with bevacizumab, that might be the case, but we now know that pembrolizumab doesn't work in mesothelioma, so it wasn't the pembrolizumab that was helping these people live longer. It was something about them intrinsically that meant they probably would have lived longer anyway. So the people in the clinical trials are not representative of the wider population, and that means that sometimes what comes out of the clinical trials in terms of numbers doesn't translate directly into what we see in real life clinical practice. And again, an example of that was the pemetrexed and cisplatin trials from 15 or more years ago. In the trial, about 40% of people responded to treatment. But then when they looked at the real world prescribing data a couple of years later from America, they found that the response rates were actually only half that, so around 20%. So it just shows that what comes out of the clinical trials, if you use this perfect population, when you translate it to the real world, which is infinitely more interesting and messy, it doesn't always pan out the same way. So one of the aims, and in fact the primary aim of, I'll just skip through these slides, but this is essentially UK data saying exactly the same thing. This is from Mesothelioma UK, showing that the median age of people diagnosed with mesothelioma in the UK at the moment is 76. The MAPS trial, one of the eligibility criteria was to be aged less than 75. So that's automatically half of the UK population that wasn't suitable for the trials. And again, the circle on the right is performance status, and it's very common to have a performance status of 0 or 1 as a key eligibility criteria for trials. So again, that's 50% of our population who wouldn't be suitable for those trials. So yeah, one of the key aims of Assess Meso was to create a cohort that was representative of the real world, of everyone who came through our clinic doors with mesothelioma, and all types of mesothelioma, any method of diagnosis, everyone. If you can come through the door and you can sign a consent form, then we want you in Assess Meso. And the aim was to follow these people up on an individual basis from the point that they received the diagnosis until they either sadly died or chose not to carry on in the study. The next aim of the study was focusing on biomarkers, and we've heard a bit about how challenging mesothelioma is to diagnose, not least because you have to demonstrate invasion on, and that usually requires a tissue biopsy. At present, we don't have any suitable biomarker, blood test or pleural fluid test to replace that. And then once the diagnosis has been made, monitoring is even harder. And this also relates to the growth pattern of mesothelioma in that it grows like a sheet rather than as a solid tumour. So it's harder to measure on scans. It needs three-dimensional mapping rather than just measuring in 2 dimensions. And although we have these 2 techniques, modified RECIST and the TNM stage, they don't correlate particularly well together. So you can go up from a T1 tumour to a T4 tumour over the course of 2 scans, but your modified RECIST could still stay the same and call it stable disease. So there's challenges there. What we do know about biomarkers so far is from Selena's very comprehensive diaphragm study, which looked at these 3 biomarkers, mesothelin and fibrillin-3, which are both glycoproteins expressed on mesothelial cells and overexpressed in mesothelioma, and then the SomaScan, which is a proteomic process. And they tested these baseline biomarkers in over 600 patients, did some mapping, some quality control stuff, and then mapped it to their final diagnoses at the end. And what they found was somewhat disappointing. The SomaScan, the proteomics, couldn't differentiate mesothelioma from non-mesothelioma pleural disease, although it could differentiate mesothelioma from asbestos-exposed controls, but that's not really the population that we're seeing in clinic. Fibrillin 3, there was a difference in values between people who had mesothelioma and those who didn't, but the threshold value, the cutoff value, couldn't be validated. And you can see from these ROC curves that the sensitivity was barely better than guesswork. The ROC on the left is meso versus non-meso, and the one on the right is versus asbestos controls. And then finally, mesothelin, marginally better than fibrillin 3, particularly when you look at the ROC curves, but still not sensitive enough for it to be used as a diagnostic test. And this correlates with the existing data, which David Arnold from our group in Bristol synthesised a year or two ago in a meta-analysis and showed the same thing, that mesothelin is not sufficiently sensitive to be used as a diagnostic tool. So this leads on to the second aim of Assess Meso, which is to create a biorepository of serial biological samples, so blood and pleural fluid, taken not just at diagnosis, but at periods throughout the patient's mesothelioma journey, and to use these to investigate biomarkers. Then finally, the final aim was about clinical trials. We know that mesothelioma has quite wide geographical variation based on areas of heavy industry and occupational exposure. And there's also variation in where the trials are held, but the two don't often map up. The trials are usually held in big cities and in places with tertiary referral centres. That said, we know people are willing to travel to join clinical trials, and it's just a question of bringing those two things together, the enthusiastic participants and the dedicated researchers, and allowing us to bring those together and move research forwards. And we know, and it's great to hear, that the Scottish Mesothelioma MDT includes the clinical trial parameter, because that definitely increases engagement and involvement in research. So the third stated aim of Assess Meso was to be a resource for future clinical trials and other studies, and to try and match up those researchers and participants. So what are the details of the study? Well, like I said, anyone can go in, any form of mesothelioma, any type of diagnosis. Essentially, if you're seen in a hospital that is open to the study and you can sign the consent form, then you can go in. Once you're in, you have a clinical assessment. You have blood tests, and including samples being stored and frozen for the freezer. Some radiology, whether that's X-rays, ultrasounds, or CTs, and then we are also collecting patient-reported outcome symptom scores, quality of life questionnaires, that sort of thing, to really get a sense of the patient experience. We're open in 15 centres across the UK, marked by the red spots, and then we've got 3 more in setup marked in the green spots. So we're gradually getting coverage. And here's our recruitment curve. We were all going very nicely until March 2020, when COVID happened and recruitment stalled, where it was suspended, in fact. But since things have been open again, since at the end of last year, things are picking up. And whilst we may not get fully back on track, we hope we're getting there. As for results, well, I can't tell you yet, because the point is we're following people up over time, and happily, in many cases, that means several years. So the data just isn't mature enough. But what I can say from looking at our demographics is that I think we have achieved our aim of having a representative sample. It does correlate quite closely with the Meso UK audit data. So that is satisfying. So then, just to finish, I'm going to run through a couple of the sub-studies and the trials that we're embedding within Assess Meso, and show you kind of what it can be used for as a resource. The first one is the TILT trial, which was my PhD. This is the trial of intrapleural bacterial immunotherapy. This was based on this research that we published a few years ago that shows, we saw from observational work that people with indwelling pleural catheters where the catheter got infected, they appeared to live longer than people who didn't get pleural infection. And that was particularly evident in people with mesothelioma compared to other malignant pleural disease. And this taps into a hypothesis that's been around for centuries, in fact, that bacteria, as very potent stimulators of the immune system, essentially could be a form of rudimentary immunotherapy against cancers. So we took that idea and we ran with it, and we developed a randomized controlled trial, which was embedded within the assessed meso cohort. And we essentially squirted bugs, or we randomized people to have usual care or to have one of two bugs squirted down their indwelling pleural catheter. One of those bugs was dead heat and penicillin killed Streptococcus pyogenes, and the other one we borrowed from our colleagues in urology and took some BCG, which they use in bladder cancer, and squirted that down indwelling pleural catheters. The trial was a feasibility trial to see whether we could do this thing and whether it was safe to do so. So it wasn't powered for efficacy. I can't tell you whether it helped people live longer, but what I can tell you is that it definitely did create both a local and systemic inflammatory response. You can see from the graphs on the left that there was a clear rise in CRP and platelets immediately after the investigational medicinal product was delivered. And then on the right, these are participant-reported symptom scores for sweats and fevers, and you can see there was a rise in their fevers, which was sustained for a good couple of weeks after the bugs were administered. So could this lead on to a larger trial? Well, on paper, yes, it could, theoretically. I think in reality, we've been trumped slightly by the systemic immunotherapy drugs, which are a bit more fancy and proven to be effective, at least, which this is not. So that was one trial. The other study, which is ongoing, I've talked about mesothelin and how it's insufficiently sensitive as a diagnostic tool, but what about as a serial biomarker, as monitoring or response biomarker? This again is some data from David Arnold in our group a few years ago. The people on the right of the screen, the black bars are people who had progressive disease, and you can see that essentially all of them bar one had a rise in their mesothelin in association with their progressive disease. Now, there were a few of the stripy bars on the left, 5 or so, who had stable disease but also had a rise in mesothelin, so it may not be absolutely perfect, but this was a small study. There was only 40 odd patients, and so we're going to try and reproduce this with assessed meso data using several hundred patients. Another study, which is being led by the palliative care team in Sheffield, is focusing on pain. We know surprisingly little about symptoms in mesothelioma. We know the symptoms that are the most common. We don't really know who they affect and how commonly they affect people. And we don't really know if they change over the life course of the disease, or indeed what they mean. So there is a suggestion that people who have chest pain at presentation may live less long, and that may be because they have either invasive disease at the outset or they have a more aggressive tumour type. But we don't know, and we don't know if the chest pain comes and goes, what that means, does it present later in life, and should that be a sign for us to get the palliative care services involved. So this is a study being looked at by Sheffield. We're also doing some genetics. We've heard already that BAP1 loss and CDKN2A loss are standard tests, histological tests on biopsy samples. But biopsies are invasive and can be a bit painful and require procedures. How's about if we try and do these tests on pleural fluid samples, which are much easier to extract from people, much less painful, much less of a palaver. So this is work being done by Beth Sage in Inverness. And then finally, a collaboration with the University of British Columbia that's been set up through Kevin and the Predict Meso network is looking at circulating tumour DNA in blood, both as a diagnostic tool and also as a response biomarker. This is reasonably early phase in that we're looking to validate the methodology first to see that we can detect this DNA at all. Mesothelioma is generally considered not particularly exfoliative, but I think their team are quite confident they'll be able to get something from blood, and then mapping that with assessed meso samples to see whether it does track progression and response. And then here, a picture that you've seen before, just summarising Kevin's Predict Meso network and really incredibly ambitious project, the main focus of which is about precursors to mesothelioma, looking at the molecular, genetic and immunological changes that lead up to disease development. But I'm very I feel very lucky to be involved in this at the final work package 5, which is after mesothelioma has been diagnosed. We are following them up in Assess Meso. And it provides this really quite special opportunity whereby, as part of Predict Meso, samples are being collected on people who have pre-malignant disease, either in the form of benign asbestos pleural effusions or something similar, and they are having biological samples, pleural fluid, etc., collected before they have developed mesothelioma. And lots of those meso origins or Predict Meso sites are the same as our Assess Meso sites. So we will have a cohort of patients in whom we have samples from before they develop malignancy through to the point at which the malignancy, mesothelioma, is diagnosed, and then following them up as their tumours progress or regress or respond to treatment or whatever. And that's really quite a special resource, and I think will allow us to provide an incredible amount of information about the biological processes driving this disease. So I'll finish there. My countdown has literally just hit 0, which never happens. So I'll summarise just to say that hopefully I've persuaded you that Assess Meso is quite a valuable resource and will shed quite a lot of light on both the natural history of mesothelioma and its evolution. I am hopeful that it does provide representation and documentation of data on groups that were otherwise slightly neglected in the literature, and also that it has huge opportunities for collaboration and translation of preclinical work, all the very clever and complicated stuff that Daniel and other folk are doing in the labs, allowing that to be translated into the real world setting. So thank you very much for listening, and I guess we've got a Q&A session.